After reading this module, you would be able to understand the following features of enzyme inhibitions. Enzymes can be inhibited by various mechanisms, both reversibly and irreversibly. Reversible inhibitors are small ligands which alter enzyme conformations in their presence, and these effects are reversed when they are removed or reduced in concentration. Regulatory enzymes are biologically controlled by these ligands in vivo. Irreversible inhibitors are used as antibiotics and can knock off enzymes. Let me introduce you to this module. Although enzymes help to speeden up reactions, one of the unique features of biocatalysts or enzymes is that they are subject to regulation. This involves increasing or decreasing their activity as and when the cell metabolism requires. Under some conditions like infection, antibiotics act on selective enzymes which are essential for the pathogen's survival and remove the infection. Enzyme inhibitions can be broadly divided into reversible and irreversible inhibition. In reversible inhibition, the molecule or ligand which inhibits the enzyme can be removed by dialysis, thereby restoring the enzyme activity. Irreversible inhibitions are created when molecules such as some antibiotics covalently conjugate to the enzyme, and this process cannot be reversed. Other irreversible changes, like in the activation of enzyme precursors during digestion within the small intestine, prevent digestive enzymes from destroying the tissue or cells in which they are made. If the mechanism of enzyme inhibition are well studied, it will be useful to design new antibiotics and molecules for selective purposes like combating drug resistance and microbial infection. Now let us consider reversible inhibitions. Many small molecules, including metabolites made during cellular metabolism, can feedback inhibit the enzyme. This is useful because needless product is not made. Furthermore, when the concentration of the inhibitor is reduced, the enzyme regains its activity. Depending on the mechanism of the inhibition, three types are recognized, such as competitive, uncompetitive and mixed. The diagnosis of these type of inhibition are made from the kinetics of the inhibition as determined by the application of the Line-Weaver-Burke plot. The Line-Weaver-Burke plot is a double reciprocal plot of the Michaelis-Menten equation, and you have learnt about this in a previous module. Let us look in detail at competitive inhibition. The equation in a competitive inhibition are E plus S giving rise to ES, which then gives rise to enzyme and product. In another set of reaction, enzyme plus I giving rise to EI and that cannot give rise to product because I is an inhibitor. In other words, enzyme binds to either substrate S or inhibitor I, but not to both. The enzyme inhibitor complex EI is therefore non-productive. The exclusivity of either substrate or inhibitor binding to the enzyme suggests that both substrate and inhibitor bind to the same site based on their concentration and equilibrium constant of the involved reactions. In the derivation of the Michaelis-Menten equation, the enzyme is therefore partitioned into two species, free enzyme and enzyme substrate complex, or E total ET is equal to E free enzyme plus ES, or the enzyme substrate complex. In the presence of a competitive inhibitor I, the equation includes the EI complex, and therefore ET is equal to E plus ES plus EI. If the Michaelis-Menten equation is derived using this equation, the final format of the equation can be shown to be V0 is equal to V max S divided by alpha Km plus S, where the term alpha is equal to 1 plus I divided by Ki. Ki is the dissociation constant of the EI complex. If this equation is converted into the double reciprocal form of the Lineweaver-Burke equation, 1 over V0 is equal to alpha Km divided by V max into 1 over S plus 1 over V max. A plot of 1 over V0 versus 1 over S results in the linear graph in the presence and absence of inhibitor I. An inspection of the graph indicates that for a competitive inhibitor, the plot has a greater slope with identical Vmax as compared with the uninhibited reaction. The apparent Km or Km prime for the reaction in the presence of the competitive inhibitor appears to increase, as is obvious because a higher substrate concentration is required in the presence of a competitive inhibitor for generating V max by 2. Note that the substrate concentration at V max by 2 is equal to Km, which has been derived in a previous module. Competitive inhibition is rarely observed in a cell or in vivo. However, this mechanism is very important in the action of drugs and antibiotics as indicated in the example illustrated in the figure. Folic acid is an important coenzyme in number of important reactions. In its synthesis, which is occurring in bacteria, pteridine plus para-aminobenzoic acid is combined to give you pteridine PABA, an intermediate, which then combines with glutamic acid to give you folic acid. If you look at the drug which inhibits this reaction, namely sulfur drug sulfonamide, is similar in its structure to para-amino benzoic acid, which is a normal substrate for the enzyme which synthesizes folic acid. However, if sulfonamide is used, then instead of para-amino benzoic acid, it binds to the substrate binding site of the enzyme and thereby blocks the synthesis of folic acid. Because folic acid is very important in the biosynthesis of nucleic acids, the bacterium which gets inhibited with the sulfur drug dies. The figure also shows how this exactly happens. The substrate normally binds to the bacterial enzyme at the substrate binding site, giving rise to the enzyme substrate complex. The product is a combination of the two substrates as indicated in the figure and is released at the end of the reaction. If instead of the regular substrate you have a drug, then the competitive inhibition occurs with the drug and the second substrate binding to the enzyme complex. However, the enzyme drug complex cannot form the product. The structure of the antibiotic or drug is similar to the substrate to enable it therefore to bind to the enzyme, and in doing so it inhibits the enzyme. Competitive inhibitors can be also designed if an idea is there of the three-dimensional structure of the substrate. Now let us look at another kind of reversible inhibition called uncompetitive inhibition. In this type of inhibition, the inhibitor I binds directly to the enzyme substrate or ES complex as indicated. E plus S gives rise to ES, which then gives rise to enzyme and product. A further reaction takes place wherein ES binds to I and gives rise to a complex ESI. However, ESI gives no product. Again, using the various enzyme complexes found in equation, namely ET is equal to E plus ES plus ESI, and deriving the Michaelis-Menten equation, V0 is equal to V max S divided by Km plus alpha S, wherein alpha prime is equal to 1 plus I divided by Ki prime, Ki prime being the dissociation constant of the ESI complex. The double reciprocal or Line-Weaver-Burke equation would be 1 over V0 is equal to Km divided by V max into 1 divided by S plus alpha prime divided by V max. A plot of 1 over V0 versus 1 over S in the presence and absence of the uncompetitive inhibitor is illustrated in the graph. As observed in this graph, in the presence of an uncompetitive inhibitor, the parameters of apparent Km and V max alter. The lines also in the presence of an uncompetitive inhibitor are parallel. Let us look at another kind of inhibition which is observed, namely mixed inhibition. Many biological inhibitors are mixed inhibitors as they appear to show some features of competitive and uncompetitive inhibitors. It is useful to point out that mixed inhibitors easier to reverse because very high substrate concentrations are not required like that observed in competitive inhibitors. It is worthy to note that antibiotics which are frequently competitive inhibitors behave to some extent as irreversible inhibitors because very high substrate concentrations are required to reverse these. In a cell, most metabolites including substrates do not show much flux in their concentration. Hence, enzymes can be regulated by concentrations of inhibitors and substrates prevalent in the cell. The various enzyme species in the presence of a mixed inhibitor are therefore ET is equal to E plus ES plus EI plus ESI. Note that mixed inhibition has the features of both competitive and uncompetitive inhibitors. The reactions leading to the formation of these various species of enzyme are E plus S giving rise to ES, which then breaks down to give you E plus P. E plus I gives rise to EI, which therefore results in no product. ES also can combine with I, that is a mixed inhibitor, to give rise to ESI. ESI also does not give rise to a product. The Michaelis-Menten equation can be derived to yield V0 is equal to V max S divided by alpha Km plus alpha prime S, which on taking double reciprocals on both sides leads to the Line-Weaver-Burke equation. 1 over V0 is equal to alpha Km divided by V max into 1 over S plus alpha prime divided by V max. A plot of 1 over V0 versus 1 over S in the presence and absence of the mixed inhibitor is illustrated in the graph. An inspection of the graph indicates that the slopes of the lines increase with inhibitor concentration and the lines intersect at a point left of the 1 over V0 axis. This type of inhibition has the features of both competitive and uncompetitive inhibitions and is also referred to as non-competitive inhibition. Non-competitive or mixed inhibition is frequently observed in enzymes, particularly enzymes which can be regulated. Now, let us look at a different kind of inhibition which is known as irreversible inhibition. In the case of irreversible inhibitors, the inhibitor generally makes a covalent bond with one of the amino acid side chains within the enzyme protein and in doing so inhibits the activity of the enzyme completely. Because the nature of the interaction of the inhibitor is covalent, it cannot be reversed. Let us look at one category of irreversible inhibition called affinity labels and suicide inhibitors. In the case of affinity labels or suicide inhibitors, these bear structural similarity to the regular substrate except that when the enzyme binds to them, an active functional group on the inhibitor covalently interacts with a nearby amino acid group, thereby inhibiting the enzyme. The covalent bond between the enzyme and inhibitor can be with an amino acid residue actively involved in the reaction mechanism, or alternatively with an important residue which denatures the enzyme. Because the inhibitor resembles the substrate, the term affinity label is used, and in more popular terms, suicide substrate, probably referring to the case where in a suicide a medicine is voluntarily or drug is voluntarily taken by the person. In the figure, two examples of affinity labels or suicide inhibitors are illustrated. TPCK, which is tosyl phenylalanine chloromethyl ketone, resembles the natural substrate for the enzyme chymotrypsin, a digestive enzyme which hydrolyzes peptide bonds in proteins following aromatic residues. TPCK has an active chloride group which, on binding to the enzyme, covalently alkylates an important residue, leading to the inactivation of the enzyme. Another example is the affinity labeling of a serine residue of the enzyme acetylcholine esterase with DIPF. DIPF is the abbreviation for diisopropyl fluoridate. DIPF selectively alkylates a serine residue. As this residue is important in the enzyme mechanism, the enzyme is thus inactivated. Interestingly, DIPF and its analogues are part of the composition of mosquito repellents. The acetylcholine esterase in the mosquito brain is inactivated by use of these analogues. Fortunately, the kind of analog used in mosquito repellent has no effect on the human acetylcholine esterase. Let us look at another kind of irreversible change which enzymes experience, and that is the activation of zymogens. Many enzymes are synthesized as inactive precursors. For example, protein hydrolyzing enzymes known as proteases, if synthesized active, can destroy the tissue in which they are made. Pancreas is the organ which synthesizes a number of the proteolytic enzymes. If the enzymes were active, they would destroy the very tissue of the pancreas. Therefore, they are synthesized as inactive proenzymes or zymogens, and when introduced into the small intestine during digestion are activated irreversibly by covalent changes, or this is also known as in situ activation. The figure illustrates the activation of digestive enzymes in the small intestine by selective covalent changes. The zymogen form of the enzyme is acted upon by specific regulatory proteases which are released in the small intestine. These recognize the zymogen and cleave at specific activation sites, thereby releasing the active site. In the duodenum, which is part of the small intestine, the pancreatic zymogens which pour into the duodenum are acted upon by an enzyme known as enterokinase. Enterokinase acts upon the zymogen form of the very active proteolytic enzyme trypsinogen 2 and converts it into trypsin. The active form of trypsin can now act further on other pancreatic zymogens, thereby converting them into the active form. This way of activating enzymes, which are known as in situ activation, prevents the wrong activation of enzymes in the organ from which they are synthesized and thereby only renders them active at the site where they are required. Let me summarize what you have learnt in this module. Enzymes can be inhibited reversibly and irreversibly. In a cell, enzymes can be bound reversibly by small ligands or metabolites to regulate the activity of the enzyme. Depending on the type of inhibition as diagnosed by the Lineweaver-Burke double reciprocal plot, inhibitions can be classified as competitive, non-competitive or mixed. Antibiotics and drugs generally are competitive inhibitors which knock off the enzymes as they are generally used at a high concentration which cannot be overcome by the substrate concentrations prevailing in the cells. Irreversible inhibitors such as affinity labels or suicide inhibitors act as poisons and destroy the enzymes they act on. Some irreversible changes occur on proenzymes to keep them from acting prematurely, like the digestive enzymes which are activated only in the small intestine.